is there. So just a little bit about my formal background. I'm Dr. Robert Westra. I'm a veterinarian. I graduated from Washington State in 2008. Before I was a veterinarian, I was a wildlife biologist for 10 years, hence why as a Peace Corps volunteer, I wanted to go to Africa. Made perfect sense to me. But in order to progress in wildlife biology, you need graduate work to kind of make it a pay for a living, because as much as they like to ship you off and do all these crazy things, eventually you do need to eat. So I went and I got a master's degree in natural resource sciences here at Washington, or in Washington State. The focus of my study was endangered species, of which a part of that, I wish it was my part, but it wasn't, was looking at the loss of genetic information when species populations get down to the scary level. So when you ask me questions about genetic diversity, it's something that's very near and dear to my heart. Most of what we have or purport when it comes to genetic diversity is based upon, so when we talk about the effects of inbreeding and the loss of biological vigor and things like that, we're talking about populations that are down into the less than a few hundred. So keep that in mind. Sometimes, and I don't know where the Ridgeback is nationally or internationally, but again, that's why I stress that it's important that we maintain the largest breeding population that we can, so that we don't deal with the effects of lost alleles and such. When I was a vet, the smartest decision that I ever made was to marry my wife. Okay, I met her in vet school, and like I like to tell people, she's the smart one. Okay, she has more degrees than a thermometer, and as a result, somebody had to go out and work for a living while she continued on. She's board certified and has all these fancy, things behind her name. So when I left vet school, I went on to small animal practice. I was in a general practice in Central Washington for a number of years, and then as I followed her to a residency, the easiest way to get hired is to go work the ER. And when I started working the ER, I realized I had a passion for it, because it's really kind of speaks to my nature: problem solve, fix it. So then I kind of transitioned to ER, and I worked in the ER for about several years. I'd say collectively, I have about 12 years of experience in small animal medicine. I started with Paw Print Genetics, which was a small genetic company, as I show in the picture here, that's like one of our booths at a show. In 2020, and I was given the mandate to come out, and education was going to be my focus, which I love. We were acquired by a company called NeoGen in 2021. And who is NeoGen? They were founded in 1982 primarily as a food safety company. They provide all sorts of tests that look for contaminant in food processing. So how does this apply to companion animal genomics? Well, in the food safety business, they also started looking at the genetics of agriculture and how to produce cows more efficiently and effectively. And then by extension, when they acquired Paw Print, because NeoGen is the largest animal genetics company in the world, with the focus, as I bolded, they want to allow customers to produce more efficiently and effectively than they could before. That kind of plays into the companion animal side. So we liked the fit because we were a small company that felt limited by our size and the ability for us to have a bigger impact on the industry as a whole. Being acquired by NeoGen has allowed us to retain what we thought was important about Paw Print, and it also gives us the capability of hopefully being able to do so much more. All right, so a little bit of an overview. I don't like doing overview slides, but what I want to talk to you today, again, when people say, Dr. Westra, we want you to come do a genetics talk, and I'm like, well, what do you want to know about genetics? And they're like, I don't know, just stuff. And I'm like, genetics is a big topic. There's quite a lot that we can talk about here. So I'm left with trying to take something that's very, very broad and make it specific and focused for you. So again, that's why I appreciate your questions, but I am going to go a little bit through the nuts and bolts of genetic testing, mainly so for those of you that don't do genetic testing to understand a little bit more about how it works and how it can be used in your program. Then we're going to look at specific genetic concerns for this breed, for the Rhodesian Ridgeback. We're going to go through the disease tests that we know about and talk about some of the concerns and the questions like degenerative myelopathy. And then there came through back channels that there's a large question, which there should be, about this condition called dermoid sinus, which affects a significant percentage of the Ridgebacks out there. So we're going to talk about what we know about dermoid sinus, and more importantly, what we don't know about this condition. And then we're going to fall back into like, so you've done genetic testing, because there was an idea that some of you would have already done some of the testing that we offered through the company for the Ridgeback. So we can talk about how to interpret results and, more importantly, how to apply those results. And this is where hopefully we can get into a conversation, especially when the information is imperfect. And this is talking about risk assessment and how to use that type of information. And then I think we've already had the conversation about balancing priorities. So let's take it way back. This is high school for me, probably junior high for some of you. But what are genetics? And this is a simple question, but it's amazing. This that you have to think about is within most animal cells, there is a nucleus. And inside of this nucleus, there's this big spaghetti of DNA, the genetic material that controls all of how life is encoded. Now, I like this slide because it shows you that at specific point in time in the cell's life cycle, that DNA organizes itself into chromosomes, okay? Trying to provide definition for words that you've been heard bantied about. So a chromosome is simply an organization of DNA. And then if we unspool that chromosome, as you can see in the slide, eventually we get down to the sequence level, all right? Inheritance happens here. These chromosomes will then split and divide into two daughter cells. And then when we talk about the evolution of gamete, not evolution, but the construction of gametes, sperms and eggs, those chromosomes will have themselves so that half of the genetic material comes from mom, half of the genetic material comes from dad, all right? But what's important down at this level where the code is, that's where mutation happens. That's where variation occurs, which can have either positive effects, like out of nowhere, suddenly this dog has got this really cool line going up its back, to, oh my gosh, why is that same dog deaf? All right, now we talked about didn't come through. We talked about chromosomes, all right? Dogs have 39 pairs of chromosomes. In contrast, humans have 23. Right? Does not mean that the dog is genetically superior to the human, although I will have you keep that in mind when you're following it after it's trying to go to the bathroom. Who's picking up what? All right? But it becomes important because this guy right here, chromosome 18, very significant impacts to the Rhodesian Ridgeback. And I like this slide in here because it kind of gives you a physical representation of what we're going to be talking about here. Now, inheritance, again, I say this, inheritance is the pass of the movement of genetic material from one generation to the next. And I'm totally blocking you, I'm sorry. It always is 50% comes from the male, 50% comes from the female. I know that those of you with children look at your children and say, that did not come from me, that wholly came from your father. I had nothing to do with that. That's not the case. The other thing you need to know about inheritance, something we learned, diarrhea is genetic. It runs in the genes. Ha, ha, ha. Love dad jokes. And insanity is inherited. You get that from your kids. Yes, and do I collect genetic dad jokes? Yes, I love them. All right, so a little bit about the mechanics. How does it work? These are all of the tests that we offer are optimized for cheek swabs. So I use the phrase from slobber to sequence. All right, but one thing that paw print started with, because paw print's primary market was the dog breeder. And so one thing we accept is a multiple of sample types. What worked for your program? Most of them, cheek swabs work great. It used to be that you needed to give us a blood sample if you wanted to get an accurate result. That is not the case anymore. Cheek swab works just fine. But we also accept tissue. We love tissue. Tissue is unlimited genetic material, right? And so we're talking about tails and dew claws, something which is probably not important to your breed. But for some, but we also accept umbilical cords, which every dog breed has one of those. You just have to process them appropriately. That information is found on our website. But what's the advantage of sending us tissue is that you can start testing at a much, much earlier age than if you want to do a cheek swab. You can do a cheek swab of any dog at any age. Genetics don't change. At conception, boom, they're nailed. They're done. So it doesn't matter what age you do the cheek swab. What matters is you don't want mother's milk in their mouth when you're swabbing the inside of it. Otherwise, we're just going to get a nice genetic result of mom, or it's going to confuse the sample from the whole thing, and we'll be calling up saying, yeah, that didn't work. So most people wait until the dog's going to be weaned, which is right around 4 weeks. And then they send the swab in with mailing the sample to us and getting it back from us kind of counts for about 6 more days. Then it takes 2 weeks to process. That's the best we can do at this point in time. So you're looking at getting results at about week 7. right about the time when you're trying to determine if this is going out to a pet home, if I'm going to save this back into my program. We understand that that's what the advantage of tissue is that you can get results back with a much more timely fashion to start making decisions, especially if you're trying to determine that fate of the dog.
What's the likelihood that you have contamination of chickens if you take a swab of a dog after they've eaten kibble?
Right, which is why you're never supposed to swab a dog 1 hour after they've eaten anything. I love this. It shows when I swab dogs, you'll see the person giving them a meat treat, you know, for behaving. And I'm like, great, can you come back an hour later? Because I don't want to get bovine or chicken or fish or whatever's in the treat. So you want them to not have had anything protein based for at least an hour beforehand. We also accept blood, semen. So if you're collecting the dog, that's where you want a pellet pulled aside for genetic testing. It kind of comes to that horse and barn scenario. A lot of people will want to test the dog after they've bred the dog and use straw. We will do that. We have been able to do it, but you're pretty much, you get what you get at that point. Genetic test is going to result, but I don't know what's going to happen to the puppies. It's better to test before you breed. That's what I'm trying to say. And then we also do formalin fixed tissues, which is kind of an after the fact. The dog died. You're trying to figure out if the dog carried for something because there's a whole, it impacts your pedigree in some manner. All right. Hey, this is the slide I need you, tech guy. Okay, this is a short video to show you how the lab works.
Welcome to NeoGen Genomics, the global leader in DNA testing for animals. Each day, our lab receives tens of thousands of individual animal samples from customers all over the world. Our lab uses some of the most advanced technology available to read the code of DNA, the blueprint for life itself, to predict how it influences the well-being and productivity of animals and the people who care for them. This tour provides an overview of how this process works. Imagine a family cattle operation that wants to make better long-term decisions about how it breeds its herd, or a veterinarian seeing a new puppy for the first time, or a geneticist at a leading university working on better ways to feed the world. For all of these customers, the DNA testing process begins by sending a sample from an individual or group of animals to NeoGen. When the materials arrive at sample reception, our group logs every item into our system, giving each a specific ID that follows it from beginning to end. A tool called the Laboratory Information Management System tracks the hundreds of thousands of tests underway. It records essential information about each sample and sets up the testing to reveal DNA encoded in the sample. Think of sample reception like a giant funnel, gathering many thousands of samples each day from many species of animals and plants. The next step involves extracting molecules of DNA from various species cells. The original sample material could be hair follicles, blood, tissue, or even unique items like deer antlers or cheek swabs from cats. In each case, the initial target units are the nucleus of the cells in the sample. To get to the DNA inside the cells, our DNA extraction lab prepares and processes cells from the animal or plant materials. At this stage, we shift from the visible world to work at the molecular level. In DNA extraction, we use chemistry to break down cell walls and free the DNA contained inside the cell nucleus. Special instruments isolate the DNA by binding it to extremely small magnetic beads. While the bead holds the DNA molecules, the chemistry washes away other parts of the cell or organic debris. The result is isolation of a clean, pure sample of DNA. Next, the lab staff place the small amount of pure DNA into special 96 well plates. These plates can hold 96 different animal samples and will be processed in economical batches. These steps, and the many ways that we use cutting-edge science to automate our technology, help NeoGen ensure highly accurate DNA test data, fast processing, and affordable services. The final lab steps are setting up the samples to run on advanced instruments that detect specific DNA markers and read targeted sequences of genetic code. First, we take the minute quantity of DNA obtained through DNA extraction and amplify it. This amplification process makes thousands of copies of the DNA. In most cases, the next step is to place the DNA sample onto a chip, or what is known as an assay. This is designed to detect targeted variations in DNA. Those targets help govern how traits are expressed in a living creature. There may be specific parts of DNA that are of special interest. For example, how do some cattle gain weight in comparison to others? Or what microbes are detected in a food manufacturing facility? Or even, will a puppy be at risk for a health condition later in life and need special veterinary support? There are many, many variations that are influenced by the genetics of the animal. Revealing this variety is complex. When placing DNA onto a chip, the target area is often stained with a fluorescent dye. When fed into special instruments, the DNA targets are excited with a laser beam. This reveals delicate changes in the DNA strand and begins to reveal how those changes may be expressed in life. By seeing this data, we can analyze the animal's genetic data for meaningful patterns. These steps rely on some of the most advanced technologies in the world, including those that scientists use to study personalized cancer care in humans. Our final stage involves analyzing the data generated from the DNA instruments. We carefully examine the results from the various scanners and employ complex math formulas to interpret the raw DNA. After careful analysis and quality control, we report the data to our customers. The findings help them make better decisions, whether they're breeding livestock, developing new science for agricultural or forestry products, using preventative veterinary care with pets, or protecting the safety and abundance of our food supply. NeoGen's goal is to serve customers by providing accurate, high-quality genotyping data, to deliver knowledge quickly so our customers can make timely decisions, and to serve our clients with fair, affordable prices. Each day, we come to work to improve the well-being of animals, to enhance the safety and sustainability of food and agriculture, and to explore the exciting new frontier of genomics. Thank you for joining us on this video tour. You're always welcome to visit our facilities. We love to share how the science of DNA translates into real benefits in our world.
Let's see if yep, the quicker works. You go have lunch. Thank you. Yes, ma'am. Yes, it's SNP and PCR based. And no, not yet. Okay.
It's a mutation that's next to the gene that you're looking, next to the base you're looking at. You missed.
Potentially. We can talk about.
Not potentially, definitely. Small area.
It's a lamppost issue. The light is good under the lamppost.
Sure. Yeah. Oh, hang on a second. Just pump the brakes and we're going to talk about that specifically. All right. So now we're going to talk about types of testing, and that's going to talk about breed identification, in which we're going to talk about lamppost issues. Okay. All right. So in my world, we split testing into two different types. There's breed specific, and then there's screening. All right. Breed specific is things that you get through paw print genetics. We have all of the mutations organized by the breed. And those are based upon the known association of that mutation causing the issue in that breed. You'll get the same service from UC Davis, from PennGen, veterinary genetics, animal genetics. They all do breed-specific testing. We'll talk about the advantage of that. And then we contrast that with genetic screening. This is where we take your dog's sample and we plaster it against our 328 different known, disease-causing mutations, coat colors, and traits. This would be Canine Health Check. This would be MBARC. This would be Wisdom Panel. Now, the advantage to doing breed-specific testing, and I'm going to just pump my own chest here and talk about ours, is that we are doing focused testing, which means you're going to get less confusing results. That's the problem with any screening, is that you're screening for these mutations regardless if that is known to affect your breed or not. This is very common for dogs to come back carrying mutations which are very, very breed-specific, or they come back with other mutations which are ancient mutations like DM, which we'll talk about in more great detail. Breed specific has the ability to be more accurate. It depends upon how the sausage is made and how they're looked at. Array-based technology right now is about as accurate as we can get. And then at the same time, because it is specific, because we're looking at a finite amount of data, paw print at least can offer our peripheral services based on that. And that's clear by parentage, especially if you have larger programs, paw print pedigrees, which is a way to share your results and increase the transparency of your breeding program, and then profiling, which is establishing who's your daddy. We contrast that with genetic screening. Again, Canine Health Check is our product, but this would be Embark that we're talking about here, which most people unfortunately have been exposed to because Embark is a marketing company and they're very good at it. All right, so I hear this conversation a lot. You said it earlier. Well, when I first started, I got all my dogs embarked. Why? Because you knew about Embark, because Embark's everywhere. If you go anywhere into the cyber world and type dog genetics, you'll get a pop-up for Embark. That's fine. That's what they do. They are what? Well, yes, they do a lot of search engine optimization to get their name out there, which is great, but they do not do their own testing, you know, and they even say this on their website. They explain that they use a service provider to do their genetic testing. NeoGen is the lab, or is a lab. We do all of our own testing. The thing about Canine Health Check, where it kind of beats the other ones, is that it's all based upon the technology that was developed through our paw print product, and therefore it has the advantages of being diverse. We're able to do different mutations, such as an insertion length for the Merle mutation or copper toxicosis or things like that, that you're not going to get through the other screenings. Now, which type is better? It just depends upon your priorities and your program. If you are working with a breed in which you are looking at a finite number of known mutations, the Rhodesian Ridgeback, there's nothing wrong. I mean, breed specific is perfectly fine and capable. In fact, that's the way I would probably push you if you asked me which one. But if you're working with poodles or dachshunds or French bulldogs, I would probably push you towards the screening because you have other priorities that are probably color related or trait related or something like that. And when you start to do breed specific testing for what I call those full palette breeds, it can get kind of expensive. So it might be the more economical option for you. But you're welcome to do either depending upon what your priorities are. Now, let's talk about SNPs, all right? Breed identification. Currently NeoGen does not offer breed identification. Paw Print never offered breed identification because we didn't want to go there. And the way it works is that within the genome, there is a certain number of known genetic mutations that are found within greater than 1% of the population, of all population of dogs. And these are the SNPs, or single nucleotide polymorphisms. That's what she was referring to. And a SNP effectively is the "you are here." It provides, I use the analogy, it's a roadmap to where you are. Now, there's two ways to use a SNP. Either a SNP points you saying you're here and you can find the causal mutation for the thing, which means if you're testing for the direct mutation that is known to cause the issue, whether that's a color or trait or whether that's to cause a disease, that is about as accurate as you're going to get. Because when you run the genetic test, we are looking for that variation in sequence, which we have determined to be the cause of the problem. Versus what I think you were referring to is what's called a linked analysis, which means we do not know what the cause is, but we know that the closest roadmap is right here. So if you find that roadmap, you found the problem, right? There is an inherent assumption that is wholly wrong, right? Yes, we can say statistically that is true, because most often if you have the roadmap, if you have the SNP, you are likely to have the causal mutation. Now, you can see the assumption and the breakdown in the logic, which is why I wholly agree, and I'm glad you guys brought that up. The problem comes is that for a lot of the disease-causing mutation, we don't know the actual sequence. So the best we have is a linked marker or a linked analysis. Okay, and again, statistically, it's better than nothing. All right, where we run into problems is in how it's marketed and presented to the consumer. All right, hey, we've got this new test for X, Y, and Z. No, you don't. You have a linked analysis for X, Y, and Z. Now, I'm not going to throw any company under the bus, but when you're a good marketing company, you're good at throwing out all these tests in which you don't really know that the test you were offering for early onset deafness is not actually the causal mutation. That got found by the same company a few years later. Hey, we'll go back and we'll retest all of your dogs for you for free. You see where I'm going with that? Does that answer your question? Okay, all right. Now, when it comes to breed ID, because it's based upon the "you are here's," the known maps of it, they basically apply this to a database and they say dogs with this breed tend to have these road markers. And if your dog has all the same road markers, we're going to say your dog is a Rhodesian Ridgeback. The problem comes is that there's genetic drift, there's variance, there's all sorts of things which could say that your dog also carries markers that aren't found within that greater percentage of Rhodesian Ridgebacks. This is why if you did a breed ID and it comes back that your dog is 25% Chihuahua, and you're like, what the heck? That has nothing to do with the case. What it means is that the test found that your dog carried the road markers that are typically associated with the Chihuahua. Doesn't mean your dog is not a purebred dog. Just means that that's what the test found. It's based upon how accurate the database that they applied the UR-HEREs to, the roadmaps to. Now, these things started coming out in big detail, like Wisdom brought theirs out 2013, and then Embark came out in 2017, and so they've been rapidly acquiring more data. People telling them that they're wrong, people sending in their own breed. So these databases are getting better. And as the databases get better, they will get more accurate. And so I say that it's not a perfect matchup, and nor do I think it should be called breed ID. It's more or less, this is what your dog carried. This is what we found, and this is what it has been associated with. Yes, I'm a veterinarian. I can speak out of both sides of my mouth with a straight face. All right. Now, that being said, when we do breed ID, we have to be careful in how we apply it. My biggest question with breed ID is, what is its function? You guys know it's a Rhodesian Ridgeback. Why? Because it came from 2 Rhodesian Ridgebacks. That is the definition of a purebred dog, is that it came from 2 known purebred dogs. That hasn't changed. That's not going to change, I hope, anytime in the near future. I want to step very carefully about a genetic test to determine whether a dog qualifies as a legitimate breed or not. Okay? Because of the inherent inaccuracies in a breed ID test, we may be disqualifying dogs that are important to conserve, and at the same time as we may qualify a dog that should not be important to conserve. So I say the question is too complicated for it to be solved at this point with a simple test. But I will tell you this, that is where a lot of energy is being pushed. So again, a purebred dogs mean any dam or sire of the same breed that are also purebred produces a purebred dog. That's from AKC. Now, registry, I guess, is more apparent to pedigree than it has to do with any genetics, and I don't want that to change anytime soon until I can be confident that the genetics really can pass muster. Now, the other thing, too, I'd say about pure breeding is that if you're focusing on your parents and you're not your puppies so much, you're setting yourself up appropriately. Because like I've said, half comes from mom, half comes from dad, and if we test before we breed, we don't have to worry about so much as to what they produce.
So, that fluidity. So you say mom and dad each contribute 50%. Is that a hard stone truth? Or can it sometimes be that a puppy holds 75% from dad because an entire chromosome got contributed and mom's got it?
Okay, so the question was, can gender or can one parent contribute more than 50% to the offspring, to summarize it? The answer is no, not to my understanding. Okay, but do you want to take us down that rabbit hole? Then why do you raise your hand? I'm going to call you out right now. We can have that conversation with alcohol involved later. Sure. Yes, sir. I have more than just that problem with linked analysis. Again, you're not wrong. Linked analyses are imperfect. I don't say that in any way, shape, or form. If we can find the causal, I mean, and there's also an argument that if we don't have the causal, should you be doing the genetic test? There's arguments to go on both sides of that equation. And you bring up the loss of alleles as one of them. So you're not wrong in that. All right, so let's bring this back down from the pros and the cons of genetic testing to a little bit about what we know and don't know about this. And this slide here, I want to say this is why it's important that I'm here, and it's really important that you're here, okay? Because breed clubs play a role in how this all works, all right? I love OFA. I love their chick certification programs. I always have problems because I get the call all the time. Do I really need a test for that? OFA doesn't require it. And it's fine. Well, where did OFA get this? From you guys. You're the ones that went to OFA saying, we have a problem and we need it fixed. So OFA listens to you, which is good, right? The problem comes is that there's other two parts of this thing, is the commercial testing company, which is me, and then there's the academic research institutions. These guys come up and say, hey, we've got a new exciting test for dermoid sinus in the Rhodesian ridgeback, all right? They go to you, and you're like, hey, they've got this great test for dermoid sinus. So then we develop the test. And then 5 years later, we realize that that mutation doesn't actually cause dermoid sinus. And this whole circle starts up again. So it literally is a three-ring circus that needs to coordinate to produce. I mean, I used the CHIC program as an example of this, but this is really how we organize our tests. When people come back, why did you call that an essential test? Or why do you call that a supplemental test? When you look at paw print genetics, this is what you'll see when you'll come up with the Rhodesian ridgeback, okay? We have it separated into an essential panel and a supplemental panel, all right? What made these essential were, one, the frequency which we found the mutation within the breed, and I've published them here. And these have the inherent biases in the fact that these are based upon our testing only. These do not, I think I put OFA when I went to the OFA database, that's what's in the parentheses. So that's what's published in the OFA database, all right? Now we have this kind of minimum threshold that once we find it in a certain population, and this handles true through genetic screening, is when we see a dog is carrying a mutation that it wasn't previously recognized to have, but we find it in a greater than 1% to 2% of the population that we've tested, we pump that up into essential, because that's where we set the bar. It's arbitrary, but that's where we set the bar to say that this matters. But you guys also have a say in that, and you'll say, you know what, I'm a French Bulldog breeder, and I know that degenerative myelopathy is inherent in the breed, but studies have shown that French Bulldogs don't get DM, which I have some questions about. But as a result, we push DM down into the supplemental panel, okay? So it has to do with, one, our conversation with the breed clubs, and two, it has to do with how often we find it. Now there's this third component. What happens when a new test comes out, right? We tend to put anything new into the essential panel. Why? Because we want you to test. We want to find out, how relevant is this? Was this just simply one really bad program out there that produced some de novo mutation that they conserved? Or is this something that is truly relevant for the breed? Again, it takes a conversation with all involved parties. So these are the ones that I'm going to focus on, are DM, early onset deafness, and juvenile epilepsy for this conversation. Because we found DM, and I've had it through peripheral sources, the Ridgeback does get degenerative myelopathy. You guys don't get the pass that French Bulldogs do, okay? And the others too have been associated with the presentation. And all of these are causal mutation tests. None of these are linked markers. Pawprint tries to only use the causal mutation unless there is no other alternative. Sure, because that's a good move. No, I'm not saying that. For the record, I am not saying that at all. Never. Frenchies have their own problems. Let's not introduce a neurologic ridge on top. Although, oh my gosh, imagine what those could sell for. All right, this is the nicest picture I could get of a DM representative, just because there are a lot of cute pictures of dogs in wheelchairs, but inherently that is sad. But I found there's a company out there that put out this toy, which I thought was interesting. But we know that DM is an ancient mutation. It's found in over 88 different breeds of dog. But we're finding out that it is truly incomplete penetrant, okay? What that means is that dogs that have 2 copies of this mutation, we assume and we move with the direction that degenerative myelopathy is a recessive condition, meaning that the dog has to inherit a copy of the mutation from both mom and dad to get 2 copies. Our genotype result would say M/M, for that dog to be clinically affected. Now, we know that there are dogs out there that are M/M that do not get degenerative myelopathy. French Bulldogs being an example of it, but not every Rhodesian Ridgeback that's MM will present with degenerative myelopathy. Why? Question. Okay, I would say it's DM in an old age dog. But no, absolutely not. No, you're ahead of the game here. Nope, I knew what you were going to say, and I'm glad you said it, because the thing is with DM is the late onset disease, right? And I asked you earlier about, the average lifespan of the Rhodesian Ridgeback is what, 10 to 12? Well, yeah. So, and in today's age where we're practicing better medicine and people are taking better care of their dogs and all sorts of other things that I want to talk about, dogs are living longer, right? So now it's a question of, is DM truly incomplete penetrant, or did the dog just not live long enough to develop DM? Okay, I think that's what you were getting at with the question. And so the point is that who knows? We don't. We still know that like dogs that, like the boxer is a classic example. There are plenty of boxers running out there that are M/M that will never show DM, but of course they're only going to live to be 8 years old. Corgis are the same way, although corgis do typically show up. So incomplete penetrant is one of those things that we have to wrestle our understanding around. So I would advocate that you shouldn't necessarily remove a dog from your breeding population because it's M/M, because we would classify this as a risk. However, there is no treatment for this condition. It is fatal. It will kill the dog. So will age. Well, I guess my professor would slap me down in vet school. Age is not a disease. But the thing is that the only way of managing this condition is through selective breeding. All right. But again, that is a shaded, nuanced question, because if our breeding population is not robust enough that we can artificially select away from this, we may have to breed with dogs that are clinically or genetically affected with this to maintain the genetic diversity that our population requires. Again, it comes back to this balanced, calculated risk. It's not correlated. It is ALS. It's a sclerotic. It's a neurologic sclerotic disease, just like ALS. We often explain it like, if you know what ALS is, that's what DM is. Okay. Yeah. That's the other question. Is it truly recessive? Because there are clinical reports of dogs with one copy of the mutation have produced clinical signs. Sum total, all that, we don't know everything that we need to know. All right, there's more to be understood here. Going back to that slide of the 39 chromosomes, one of the points that I was going to make was that there's parts of the genome which we consider to be coding, which means that we know this part of the genome produces a protein, it has a specific function. There's other parts of the genome which we consider to be non-coding, and about a year ago we would call that junk DNA. But now we're realizing that it's not junk. It actually has something to do with whether or not a gene is expressed. Now, not all of it, but we're learning in that non-coding region that there are things that actually control whether or not a gene becomes expressed or not. So this is where things are starting to go in our understanding of genetics, is that these dogs may be M/M, but there is another part of their DNA which is preventing clinical signs. Or this dog that only had one copy of the mutation is presenting clinical signs because they are lacking that suppression DNA. This is wholly speculative at this point, but it tends to speak to the issue. Yeah, and so my last point is I say use caution when limiting the breeding population. That applies to dogs like the French Bulldog, but because we know the Rhodesian does get this, if you can afford to breed away from it, you should breed away from it, because that's all we can do to manage the condition.
If the sire and the dam both have, or if only one of the parents has the gene, does that mean that it's most likely the puppies won't get DM? Okay, so to repeat your question, if the sire and the dam have the mutation, or it is just one parent have the mutation. So we're talking about inheritance, which is going to come up in just a couple of slides. It's a great question. So the condition in the essential panel is the early onset adult deafness. This is a mutation which affects chromosome 18. And because it affects the gene, the EPS8L2, which affects hair growth. Again, this is what we suspect that is causing the deafness in these dogs. It's been shown that they typically affect as early as 4 months. Dogs to 1 to 2 years will present with this. The only true way to diagnose it is through bear testing. But what makes this unique is that it's not melanocyte related, which means it does not have to do with the color producing cells that it causes deafness in the what's called the lethal white dogs or the double merles. This is your own special flavor of deafness. So feel special about it. But interestingly enough, and I'm wholly on my own limb, please do not crucify me. That was what that whole definition was, but it affects chromosome 18. Your breed's affected by it. Chromosome 18, keep that in mind. Maybe, I don't know. Juvenile myoclonic epilepsy, all right, can present as early as 6 weeks. There was one case report of a neonate at 6 weeks that showed this. This is clinically presented with myoclonic spasms. Basically, it looks like involuntary muscle twitches that will progress to full tonic-clonic seizures. And it can cause whole body movement. It can also be very, very subtle. In the paper, the one thing that they talked about is the inability to tell if a dog is having absent seizures. That's the suddenly going static, because it's very hard, you know. In a human, we can see like what's wrong with you because suddenly they're non-responsive. I look at my dog all the time and she's non-responsive. But the interesting thing about this is that environmental conditions, I mean, there is a strong correlation with photosensitivity and this mutation in that one of the studies showed a flashing light and was able to induce the spasms with that, which presents a really unique treatment opportunity and gives whole like relevancy to the product called Doggles. Specs for dogs, exactly. And this is me again on my own limb here because I just want to see more dogs wearing sunglasses. I think that's cool. But one thing I thought was interesting is that in the case, some of the dogs were euthanized due to progression of seizures and lack of response to treatment, while other dogs live with this mutation throughout their whole life and had a perfectly high quality of life to live with. And so that brings into question, what do we do with this mutation? And honestly, because we can't control the severity of presentation, I would wholly say you need to breed away from this. Then in the supplemental panel, we have hemophilia A and B. These are both considered to be X-linked recessive, which means a dog has to receive 2 copies of the mutation because it's found on the X chromosome. We tend to find this presented more often in males than females because males only have the one X chromosome. So that becomes, and the X chromosome is expressed, so they will present with this. Now, having said that, the frequency data for these 2 mutations was very, very, very low, which means, I mean, it was like less than 0.001% of dogs that tested had it. I think we found 1, which meant that the frequency of these mutations is so low in the population as we've tested for it. The relevancy of these mutations becomes, I guess a long way of saying is you guys are doing a good job because you can affect change in your population through selective breeding. It's usually within just a few years of breeding away from it, you can get mutations out. And that's more or less what I want to emphasize with this, is that yes, the mutation exists. Yes, it does affect the ridgeback, but it's in such low frequency, it's not a very great concern for you. All right, now this is where things get interesting because I went around the horn and I checked other websites and I made some consultations with some people who work for some other companies. And there's some really interesting things out there. Is that some are recommending that we test the ridgeback for progressive retinal atrophy, affected by the PRCD variant of progressive retinal atrophy. It's an eye disease, which means that they've found an affected dog. Malignant hypothermia was suggested that some people are recommending testing for. Ichthyosis, which is a golden retriever dermatological condition that they are thinking that's effective to the ridgeback. Inherited ventricular arrhythmia. This is Kate Mears out of NC State. And I will wholly say with that one, there's more information coming. She's working on that as to whether or not it's relevant or how do we use that. And then exercise-induced collapse. And this is a common sporting dog mutation. And again, I don't know if these are important or not. I just found it interesting that it was very hard for there to be kind of a collective agreement about what we found in the Rhodesian Ridgeback. This is where the Breed Club steps up. This is where you guys compile data, you talk to one another, you have a health committee, you have a genetics committee. These are the kind of things where you can actually, apply your finger to the scale and say yes or no.
And I think that's something that's kind of an open question. This is more meant for a discussion that you guys should have, is that this is what you need to determine. Don't let the commercial industry push tests onto you until you've determined it's valuable. All right, let's talk about dermoid sinus. How am I doing on time? I mean, I'm way over, that's for sure. So, but if you want me to continue, I will. Okay. So dermoid sinus is a congenital abnormality consisting of epithelialized fistula. That just basically means an opening or tube that connects the skin to the vertebral canal. It's caused by an incomplete closure of the neural tube. The neural tube is something we're talking about way early in development. You know, the gut is one of the first things that happened, and then right along top of it is the future spinal cord develops. So this is something that happens at the very early embryonic stages. There is a scale of severity from 1 in which it's just very superficial all the way up to 6. And I think 6 only affects a certain breed. But the one that really concerns us is 4 and 5, because this is when the deep sinus connects the outer world all the way to the spinal canal. And then because this is epithelialized tissue, it produces hair and oil and all sorts of wonderful things, which collects in this sinus, which then can create a risk of infection, which is your meningitis or empyema, another bacterial infection that can happen down into the neurologic level. So basically, we're taking the outer world and putting it somewhere it doesn't belong. There is one form of dermoid sinus, which is actually a dermoid cyst, which means that it only happens within the body and it never extends to the outside world, but it's still producing hair and oil and all sorts of garbage. The thing is that most of these dogs are going to be aclinical, meaning you won't know that they actually have it unless that cyst translates to the outer world and then ruptures and you have a disgusting mess on your hands. Data suggests that 8 to 10% of Rhodesian Ridgebacks are affected. That's based out of Sweden, or the Swedish population, I believe, but I don't know how it applies to the population as a whole. And then affected dogs are either cull or treatment. And the one thing I looked into the clinical treatment of this, because honestly, if you were to tell me when I was in practice, this dog has a dermoid sinus and hey, doc, can you fix that? I would literally break out in cold sweats, because there are amazing anecdotes of the clinical treatment of how they correct these dermoid sinuses, which involves very, very invasive, labor-consuming following this dermoid sinus down to some very scary levels within the dog. So we don't engage in treatment lightly. I don't think any veterinarian should. And if they're very cavalier about it, you might want to consult a different surgeon. But the other thing I saw about the treatment for this is that getting it sooner than later tend to have a better prognosis, which means if you can affect the young dog or treat the young dog much more than trying to treat the older dog in which it was just casually ignored for a period of time. But even treated dogs then should be petted only, you know, because here we're getting into more like the conversation about hip dysplasia, and that we do not know the causal mutation. I'll say that right out from the beginning. We're going to review that in a minute. We do not know the causal mutation for dermoid sinus. Therefore, the best that we're left with is breeding dogs that we, to the best of our knowledge, do not have dermoid sinus to other dogs that do not have dermoid sinuses. And as such, then we can hopefully produce more dogs that do not. Now, we're going to talk about that in the next slide. The one question I have for you guys is that in the papers that I've been reading about the progression of the understanding of the genetics of this condition, is that in the original papers that came back out in 2006 and 2007, they made the claim that ridgeless dogs are not affected by that condition. Do anybody have some personal experience with a ridgeless dog with a dermoid sinus? Nothing? Okay, because later in the publications, they talk about ridgeless dogs. There was one or two in their population study that were ridgeless that did have this condition. And I think it's still under debate as to what it was that caused it. Correct. Right.
And it's been tagged to us because it is so obvious.
Mm-hmm.
Or is it a condition that in the formation of the Ridgeback as a breed was brought in
Right.
something at the beginning?
So here to kind of share your comment,
was because other breeds are affected by dermoid sinus, means that maybe the Rhodesian Ridgeback is being unfairly singled out for this mutation, or did the mutation come from somewhere else when in the formation of the Ridgeback it was brought in, if to summarize correctly. So let's look at the genetics that we know about. The original publication did a great job of finding the mutation which causes the ridge. Okay, they know that it's a 133 kilobase pair. That's the sequence, going back to that slide, of DNA that has a duplication. And this is found in chromosome 18, okay, which incorporates 3 genes that cause or determine fibroblastic growth factor, okay, which help with many different things, but the one we're concerned about here is they also promote hair. Question? In the studies that I said, they only mentioned the Rhodesian Ridgeback and the Thai Ridgeback. I did not see anything about yeah, correct. In fact, that is a new one. I'll have to go look that up. So thank you. Now, so that we know that it incorporates on that chromosome 18 that causes this. Now, they found a strong association between dermoid sinus and the mutation. So they were saying, yes, it is the ridge that causes this. And then in 2015, a validated genetic test was developed for that 133 kilobase pair duplication, which meant that, yay, we can test for dermoid sinus. No, we can't. Okay, because in 2022, they came out, I'll come back to this slide real quick. They came out with another paper that showed that 51% of the Rhodesian, of the 77 Ridgebacks that they tested were ridged, had a copy number risk of greater than 2, that's their thing, but they did not have dermoid sinus, which means there was an imperfect association between having a ridge and having a dermoid sinus. Okay, so they were able to say that, I mean, the collective theory is that if you had a dog that was homozygous, meaning it had 2 copies of the mutation that causes the ridge, that dog was at greater risk for producing dermoid sinus. Now, to follow it back is that because not every dog that has a ridge has dermoid sinus, we would say that mutation has incomplete penetrance, like DM. Not every dog that has a ridge is going to develop the disease. But if we assume that dogs with 2 copies of the ridge are at greater risk, well, that opens up a breeding opportunity, right? That we can reduce prevalence of dermoid sinus. For the record, that is not true. At this point in time, to our greatest understanding, again, talking out of both sides of my mouth, that is not true. The paper in 2022 came out to show that having a ridge, being homozygous for ridge, meaning capital R, did not predispose a dog for the dermoid sinus, which means that we don't know what causes the dermoid sinus. Speculation, we do not know, would be that likely the disease-causing mutation is in there somewhere, but we don't know exactly where it's at. And I think a linked marker or linked analysis at this point in time wouldn't do anybody any good. So what brings up the fact is that the only thing we can say for certain is that we know that there's a strong correlation between the breed and the disease. Just like, you know, we know that golden retrievers can get not lipomas. Well, ichthyosis, no, but I'm talking about cancer, sorry. Lipocychromas. You know, we know that there's a strong breed predilection between the breed and cancer, but we don't know the genetics behind that. That's where we'd say this, is that dogs, that Rhodesian Ridgebacks are predisposed to dermoid sinus. We do not know the disease-causing mutation. So this puts into the category of a genetic risk, right? And whenever we're dealing with risk, now we have to go back to making those calculated decisions about who to breed to what. Is it something that we wholly ignore? Not necessarily, but I think you have to have your priorities organized before you use this as any kind of mantra to say yes or no.
But you can prevent it with supplements, and people who don't have it in their parents can still get it as well.
Yep, that's why I wanted to go back to this slide, because we know that there are potentially unknown environmental factors which may contribute to it. Folic acid is one thing that they have not studied. I did not see a result for that, but it was purported as is that maybe it should be something to consider. I'm not suggesting that, saying that that's just what they talked about, is that even in 2015, after they validated the test for ridges, effectively, that's what it does. They said that because there is this discrepancy between dogs that are homozygous for the ridge and the presentation of dermoid sinus, there are still either ambiguous genetics or unknown environmental factors. So I almost feel like you wrote this. Your questions are fantastic. Yep, no, this is great. This is really great. I appreciate that. But yes, I agree with you. And that's the thing is they don't know. And so there is this suspicion that either there's unknown genetics involved or that there's environmental concerns. So moving away from dermoid, any other questions about dermoid sinus? Moving away from it, now that you got your report back, now what? How do we use genetic information? This is going to go to your point there, okay? So first of all, when we do it, we produce a genotype, and we use these two commons. WT stands for wild type, that is baseline or normal, all right? So when you see WT, we did not find the variant. We did not find the mutation. That dog has what we would consider to be a normal gene. We contrast that with mutation, which is M. So a dog can have these types of results. It can either come back WT, which means the dog does not carry the tested mutation. It can come back WTM, and if the mutation is recessive, we would call this dog a carrier, which means it itself is not going to present or be at risk for the clinical disease, but it could pass that on. Then finally, we could say the same genotype, WT is M, but this dog is at risk, all right? That means that the mutation has what we consider to be dominant, codominant, or complex inheritance, like degenerative myelopathy or something. And so we would say having one copy of that mutation means that that dog, we have a clinical concern for that dog, or there is a potential. And then finally, the dog comes back with two copies of the mutation. We would call that dog affected, meaning that for everything that we know, that this dog has two copies of the mutation and may present with the disease. So dominant versus recessive question. No, these are for the causal, when we test for the causal mutations, we're looking for that sequence of DNA. I'm not understanding your question. Whenever we do a test for a causal mutation, we're looking for that sequence of DNA in the gene that's been associated with the disease, not just a linked SNP. If we do a linked analysis, we'll mark it as such, or it's marked as a linked analysis. It is. They want to bury that because they don't want you to question their accuracy. Nope, it should. So when we're talking about dominant or recessive, this is just a way for understanding the way we write it. I'm using you guys as an example because you got an amazing example of a dominant mutation. Capital R means the dog has at least one copy of the mutation. Lowercase r means that's the recessive. In order for the dog to present with a dominant, they only need one copy. So the genotype can either be homozygous, R/R, or heterozygous, capital R/lowercase r. Either way, the phenotype is going to show. Versus a recessive, where they have to inherit one copy of the mutation from, or the recessive from the mother, one copy of the recessive from the father, and that's how we get a ridgeless dog. Is that you bred a dog that is heterozygous, meaning capital R little case r, to another dog that is heterozygous, capital R little case r, you have a 25% chance of producing a ridgeless dog. Each puppy. Potentially, yes. So recessives, we love them. And thankfully, all of the diseases, except for the dermoid sinus, because we don't know, that are in your essential panel are recessives, which means that they're easier to breed away from. And this goes to your point is that because carriers are not affected, they should not be removed from your breeding population, right? You just need to breed them accordingly. Take a carrier and breed it to a clear, and you have no risk of producing an affected or at-risk dog. So you take a normal dog, which has two copies of the normal gene, to a carrier that has one copy of the recessive, you see that the probability is you're going to get 50% carrier, 50% normal, but you're not going to produce any at-risk or affected dogs. And at the same time, you get to conserve the parent in your and hopefully got to conserve whatever feature or trait you were trying to promote in your program. No, these are great questions.
So then why are so many litters that are produced having
Because, now you're gonna make me jump slides. Sorry, but that's a great question. She asked why are so many litters of ridgeless dogs being produced? Okay, because God still gets a vote. I'll preface it by that in a moment. All right, so this is why you need to conserve your carriers, all right? Because we can still breed away from the disease and still maintain dogs within the population, which still promotes genetic diversity, all right? And as a result, is if by selective breeding away from the disease, the breed improves, meaning you're reducing the frequency, which is why we don't see hemophilia within the Ridgeback anymore. And at the same time, these tests are demoted, which means it's become simpler to deal with that breed. Now, if it's not recessive, but it's dominant, we only need one copy of the mutation. This is redundant for you guys because you see that here, you can breed a heterozygous dog affected for Ridge to a dog that's no variant. But now, you get 50% chance of getting no variant and a 50% chance of getting a Ridge. The wording is percent chance. Sorry, I'll come back to this slide because everything is a probability. All right, so if you bred a heterozygous Ridge dog to another heterozygous Ridge dog, you only have a 25% chance of getting a ridgeless dog. It means there's a 3 in 4 chance that you're going to get ridges. But you've seen multiple litters where you'll get multiple ridgeless dogs. That's because the dice came up 7 for that dog each time. It's a probability. And yeah, there've got to be other factors involved because sometimes you just see it happen. And it's like you flip a coin 4 times. You know, you should get heads twice, tails twice every time you do that. Sometimes it's going to come up 4 times heads. Sometimes it's going to come up 4 times tails. That's probability. And that's all we can tell you with a genetic test is that this is the percent chance that you would produce that homozygous recessive dog. Okay, and I'll show you here. So we use this Punnett square. All right, now remember I used WT and A. For this one, we're just going to use A. So this is a simple exercise that you can do. For those of you that's old hand, this is going to be redundant, but for someone who's new to this, this is it seems a little simplistic, but it's super helpful. All right, and that is just draw a 4 square or a big plus on a sheet of paper. Put one parent, doesn't have to be the female, but put one parent on one side, put another parent on top. All right, now I've got their genotypes. Now in this example, both are homozygous for the A locus, just using this as an example. But then we fill in the boxes. So the female's genotype for that top allele fills the top 2 boxes, and the bottom allele fills the bottom 2 boxes. I go through this in this level of detail because it's important how you fill this out so that you get it right. Take the parent on top and pull the genes from the boxes on the left to both boxes on the left to both boxes on the right. You can see how that works there. And what that does then is now you're looking at that percent chance that those 2 parents will produce those offspring. So when I say that if you breed a carrier to a carrier, one box is going to be capital R, 2 boxes are going to be capital R, lowercase r, and then there's going to be this one box that's going to be little r, okay? And that's where we get to probability. All right? Everything, if you breed a carrier to a clear, the percent chance is you got a 50% carrier, 50% clear. If you breed a carrier to a carrier, or a heterozygous Ridge dog to another heterozygous Ridge dog, you got a 25% chance of producing capital R, 50% chance producing heterozygous Ridges, but you get that 25% chance of producing the ridgeless dog. But that's a percent chance, so that's for each puppy. And then if you breed a carrier to affected, it's 50-50. Questions? associated with the
Be on presentation of that rich certificate. Talked earlier during our roundtable discussion about keeping the breeding opportunities as open as possible, but we as a breed club are restricting our opportunities by saying this puppy here that came from the same parents as this puppy that has a ridge, but this puppy here doesn't have a ridge, so I'm not going to use it, even if it might be better genetically, better phenotypically in structure. We are shrinking our genetic pool. From the genetic side, is there any research being done to determine if there are specific, other than the dermoid sinus, specific conditions and diseases that do not come with the ridgeless, but only with a ridge, either hetero or?
So the question is there any research being shown that ridgeless may have some other benefits to conserve them? Other than, right, other than maintaining genetic diversity? I do not know the answer to that question. Okay, but if you breed a homozygous ridge dog, capital R, to a ridgeless dog, what's the offspring going to be?
No, 100% carrier.
100% heterozygous ridge dogs, because the parent with its capital R can only pass on capital R. We go back to the Punnett square. The ridgeless dog has little r, can only pass on the little r. So when we fill out our Punnett square, they're all going to be capital R from the one parent, little r from the other parent. So all of the offspring are going to be heterozygous, capital R, little r. What are they going to look like? Ridge dogs. Yep, nope, that's perfectly fine. This is why we do this. This is exactly why we do this. So then I throw it back onto you, because I'm not going to sit over and say, maybe you would, and I'm not going to offend anybody, maybe you would breed a ridgeless dog because you like the color, you like the temperament, you like the conformation. It's the perfect size. It's not too big, it's not too small. Correct. Exactly. Correct. Correct. And so at this point in time, I'm going to say as politically as I can, the link is in question, right? Pretty much they've disproven it, but it's in question. You can never prove something doesn't happen. And the fact that, yes, a test exists, you could definitely do that. No, but I'm not saying that. I don't want to upset any breed standards and whatnot, but that's the thing, is that you have that ability to do that. And that does hopefully, increase the opportunities and options for somebody when they're doing their breeding. And I've said this multiple times, but here I spelled it out. You have to always manage and balance the equation. When you're trying to cull something out of your program, you have to know what good you're losing and what bad you're losing. You always have to make this balance. That's why it's important for you to come to this. That's why it's important for you to ask these questions and get educated, so that even if you're making the best decisions that you can. And I don't want to get into inbreeding just at this point in time, but this is the thing, is that you want to conserve your positive features and traits. You don't want to lose them, because I don't advocate going scorched earth. Never say, that's it, no dog with this mutation will ever come from my household. That's a little hard line thinking. It requires nuance and balance, understanding, education, and it is a challenge, but you know that it's a worthy challenge.
It's spontaneous developed, and then it was perpetuated through either dogs doing what rabbits do or humans purposely putting dogs together. So with all of these tests, how can we as breeders better prevent or know whether or not a mutation that we see is something that came through our lines or just spontaneously developed in that particular litter?
Right. No. So the question is how do we know if a breed is it's an ancient mutation that came through the development of the breed, or if it's something that developed de novo within one dog and then was conserved? And that's where genetic diversity comes into line. My initial take on your question is that by maximizing genetic diversity, you reduce the chance that a spontaneous de novo mutation would be conserved, because you're increasing diversity. Okay. At the same time, that is where there's a lot of argument that we can potentially through testing get rid of the bad and keep the good.
Know what the parents have, shows up in the offspring, you know, it's spontaneous. Parents know that. And we can make a better genetic decision on
Once we
by
get,
doing, by testing, when it's cost effective, test every single dog.
and once we get to the point where all of our tests are positive. Yeah.
So in whippets, there's a problem with people in costing in other, you get better quality. So every whippet requires certain parts of testing genetics. So you can improve the offspring on also. And it just,
I think it's interesting that are you advocating for that or for disease testing or for breed identification?
well, making sure the parents match the kids.
Okay, well, then we're talking about profiling, and you and I are wholly on the same page with profiling. I'm absolutely. That's correct. Yep. So
Spaces and with the
So people actually, breeders actually do that,
Yeah.
yes, yes, right. So
If you have a kennel with two intact dogs, and you don't realize that one of them got a hold of your bitch, and you purposefully bred to the other dog. Now you've got a litter of puppies that you think came from dog B, but if you were to actually do a genetic roadmap, you would find out that dog A was the sire of that litter.
Yep.
All we have is the sheet of paper saying this is the dog I bred.
So let me stop. Let me interject right here real quick. Just let me put it. Not just a genetic roadmap, parentage. Parentage is a different type of test than breed ID. Parentage is we looked at your dog and we developed a fingerprint for your dog. And we did that to the father, then, you know, for both sexes. Then when we look at the puppy, we can see up to a certain delineating point, this dog shares the same genetic fingerprint. That, right, the who's your daddy, right? When we talk about parentage, absolutely. I mean, and that's to me, like I want to see genetic parentage or profiling being done in the same way that we do microchipping.
Test both every time at the same time.
Do both.
Checking
Right. So a bundle that does,
Yeah.
that does,
doesn't have to solve.
right.
And it's the problem with how eggs.
It was an accident, or are we talking?
Not always.
Oh.
Sometimes.
So that's maybe this is to your point, like you're talking about a bundle where it's all included. And I will say that AKC is moving in that that very direction.
They
So
have two different types right now, satellite test versus the other, what is it called? That if you stored a dog 20 years ago, they went to a different platform, AKC.
right, that just has to do with the shift in technology, right?
Dogs.
One bitch, and then the puppies.
Dual sire
Yeah, you do dual-sided litters. And genetic testing will tell you who is the father of that dog, even in a dual side. In fact, that's where it often is used.
Dental litter where you thought that this dog was bred, but that dog actually got a hold of the bitch. Or to your question, someone unscrupulous could say, I bred this dog, he's the sire of this litter, when really that dog's throwing blanks and they use that dog, but they want people to think it's the sire.
Yeah.
Right.
Has happened.
Yep.
Wow.
Yeah. Never let people's ability to
Good to see you.
have a different set of priorities.
Or
Is that,
Yeah.
I guess I want to leave you on this point is that because you are producing dogs, is that we've seen a shift in the dog population, that they went from the barnyard to the backyard. Now we see that over half of people who own a dog, let that dog sleep in their bedroom with them at night. I got called out by one of my colleagues because I used to say, let them sleep with them at night. He's like, I sleep next to my dog. I don't sleep with my dog. Okay, the point is that it behooves us to breed the healthiest dogs possible. Because these dogs are being incorporated as family members, of which I'm sure you are just as guilty of as I am. Question from the Q&A.
Well, this one says, will there ever be a genetic test for TGAA thyroid? Are there any studies on why passing RRs produce, or capital R's produce failing offspring?
The answer is yes and no. I would assume that eventually in time, as time, research, and motivation prevails, a genetic marker will be associated with more and more. But at this point in time, there is none. And again, to the question is, we do not know, or I am not aware of any research that's looking at that direct linkage between capital R, and the thyroid condition. Thanks, guys. I mean, yeah, we hang in here. Some of you guys have been in here for like 2.5 hours now, so. No, that's quite all right. But I will say this, it's like one of those things where I'm either running out of things to talk about, and it's like, so how about them Bears? There you go. No. Nope. But I do know this is that sometimes if you give me enough room to talk, I will just keep going, because we only scratched the surface, crazily enough. Spokane. No, I'm actually heading back to Denver here shortly. But my question to you guys is, and I think you answered this for me, is that we've been talking about back and forth the color of Rhodesian Ridgeback. Okay, and I know it has nothing to do. You guys don't care about color, or maybe you do. Have you ever seen a black Rhodesian Ridgeback? Ah, you guys are awesome. Thank you for letting me be here today.
Yes. Recessives in our lineage, we do not have black.
Okay, all right, that makes sense.
No, I a true black with no. Yeah. Completely black. And it was
Unless it was an altered photo on
Okay.
the internet.
Yeah.
That
So
was
one
going
of our
around.
breeders, one of our foundation breeders has a web page where she's got actual photos of dogs exhibiting different colors that we used to have. We carry brindle, we carry blue, we carry black and tan.
Right.
We used to carry more white than we display now. And then when the breed standards were written, we narrowed it down to Wheaton with minimal white.
Okay. No, that makes, because just looking at, and I've been looking at them all morning, I'm like, I can't tell by looking at them. I mean, the ones that have a mask, that's pretty obvious. But the ones that don't have a mask, you can't tell if they're just recessive at the E locus and they look like an Irish setter red. Okay, and I'm not calling them Irish setter. It's just, or are they all the way down through the K locus? They're all homozygous KY, which means we're seeing the sable come through, which is the Wheaton, and there you can see a little bit of mask or something. But if that's the case, then if you get dogs that are lower E, little e, and dogs that are big E, then that potentially can produce black. Brindle is all in the K locus as well. That can also be a natural mutation within a dog. And brindle sucks because we can't test for it. We don't know the inheritance of it, whether it's dominant or recessive. We do know that it tends to be recessive to black, but dominant to sable or tan points. And the fact that that's agouti, yes. We're all on the agouti spectrum because your tan points are on the agouti spectrum. You know, banded hair, effectively.
We should never look like a red bone red.
Right.
We should have Wheaton in us, but we can have a deep red Wheaton that pushes into Ridgeback territory for red bone, and then we can go all the way to dead grass, which is almost blonde or near white-ish cream. Yeah.
Let me show you the gosh, lots of Punnett scores. More Punnett scores. Geez, think I'm a geneticist or something. Yeah, when you're on the agouti spectrum, that is your variability. I'd rather you not take a picture of that because I stole it from the paper that I cited from it. But if it shows up on Facebook, I'm dead. The answer is no. Because I did cite where I got it from, because this is Danica Banish's from Danica Banish's out of UC Davis. That's her paper, but I've never. Okay, there you go. That's true. No, we've stopped I'm not sharing screen anymore, are we?
Are we still?
Yeah, you're on.
Oh, okay.
You want me to stop that?
Yeah, that's fine. It doesn't matter. I stole it from Danica. Danica, it was your picture. Oh, and I also want to give full props to Dr. Jared Bell out of Tufts University. He can